here in Cambridge, and then did my PhD in Oxford. So a lot of you will know me through my research that I did in Oxford. And 7 years ago now, I moved to America to initially work on a kind of research project at the FDA related to cardiac modeling, and I've been involved in FDA, so US Food and Drug Administration, FDA efforts related to computational modeling for a little while. So for those of you who are not familiar with the agency, I generally try and show this slide at the beginning of my talks because FDA only has food and drugs in its name, which is a bit misleading because we regulate medical devices as well, and FDA regulates biologics such as blood supply, gene therapy, stem cell therapy, and does other work related to tobacco and so on. So to be clear, I'm in the devices center of FDA. I don't work in drugs. And another thing I do is that we recently founded a modeling and simulation working group, which has brought together people who work in modeling and simulation across all these different agencies. So there's a few thousand FDA employees across all the different centers. There's a few thousand FDA employees at FDA, and people who work in modeling and simulation either through doing research using modeling and simulation or reviewing simulation results submitted to FDA. And so we recently founded a working group that's kind of bringing these people together for the first time and answering trying to address the common questions that we have. So why is FDA interested in modeling and simulation? Certainly in the medical devices center, we're used to there being three sources of evidence that are submitted to CDRH, my center. Traditionally animal tests, bench tests, and clinical trial data, but increasingly simulation is being used as a source of regulatory evidence, and also, as I'm going to talk about in a few slides, it's being used in other ways as well. So one of the things that that this working group that I mentioned has done is, because simulation is acknowledged by the agency management as being very important, we're well past the point where management is skeptical about modeling and simulation. They understand that it's being used, it's important, it's going to change practice, it's going to change a lot in healthcare, but there was a lack of understanding, lack of information on how FDA researchers are using modeling and simulation. So one thing that we did is try and collect data by reaching out to these scientists across the agency and ask them how they use modeling and simulation, in what fields, which are the rows here, and what kind of applications areas, which include things like pre-market review of medical products, devices, drugs, post-market assessment when there might be an adverse event that occurs for a device or a drug, then FDA is tasked with figuring out what happened. So it can be used in those application areas, or modeling and simulation can be used in policy development, policy implementation, and there's an other bucket as well. So we collected that information across all the different centers. So you'll see that the center which regulates drugs is the one that uses modeling simulation in various different types of modeling domain and across all the different application areas, including a lot on policy implementation. My center, the Green Center, the Center for Devices, traditionally uses the physics-based domains, mostly in the review. And the other centers, it's a bit less, but it's increasing. And this, basically, I'm showing you this to give you an overview of how modeling is used at FDA, and I'm sure this will increase going up. So for CDRH, the Devices Center, there are three different ways that we can kind of categorize how simulation is used. It's been used for a long time to provide regulatory evidence about devices. So, for example, implantable devices, one of the important questions about those is whether they will fracture in the body. That's obviously a life-threatening event. So it's a standard approach to as well as do bench tests of those devices to see how structurally stable they are, but also do simulations with those devices, maybe to reduce the amount of testing that you do. That's very well established. That's actually very similar to the kind of the questions and challenges are very similar to how structural mechanics simulations are used in engineering. Then you have the idea of using models of patients to test these devices or to test virtual devices. So you get into the idea of virtual cohorts or digital twins or sets of digital twins. And these can be used in preclinical testing. They could potentially be used in clinical trial augmentation, which is one thing that there's a lot of work being done on at the moment. There's people trying to develop a statistical framework for using digital or simulation-based data to augment a real clinical trial. And these images are from something called the virtual family, which are anatomical models that have been used also for a long time to provide evidence for whether it's safe to implant whether it's safe for a patient who has a metallic implant to undergo MRI, because those implants could heat and cause significant damage in MRI. So to obtain labeling, to obtain the ability to label your device as MR safe, then what people have done for a long time is you can't run clinical trials, but what you can do is do simulations on anatomical models, solve Maxwell's equations, very well established, and try and predict the heating. Then the other way that simulation is now starting to be used, and the first one is the HeartFlow device, which some of you might be familiar with, is where you go in and use imaging data of the patient, and that is used to make some patient-specific to do a patient-specific simulation and make some predictions. The HeartFlow device provides information about coronary blockages of coronary arteries. So this is a completely different type of application. This is now not where the model is actually the device, and we call that software as a medical device, or similarly, that you can have software in a physical medical device. And so there's various different regulatory questions related to those, and we have this challenge that we've heard about: how do we know how to trust a computational model? So that kind of gives you a motivation for where I'm coming from when I'm asking this question. FDA, we don't develop the product, but we have to evaluate the product. So we're trying to think about what the questions we should be asking, drawing a lot from what has been done in engineering, the verification and validation literature, or the VVUQ, verification, validation, uncertainty quantification literature. However, there are unique challenges associated with using. We can't just take what's been done in traditional engineering and just apply it to healthcare, because there's many unique challenges. For those of you who are not familiar with the terminology, verification is about confirming that the numerical solution has been a correct solution of your mathematical model, and validation is actually checking if your model is a correct representation of reality in some way. So at the end of this talk, I'm going to talk a little bit about uncertainty quantification for cardiac models, but before that, just a very quick overview of some other work that's been done by the devices community to get you a little idea about where we are and the bigger picture. So ASME, the American Society for Mechanical Engineers, has for several years been putting out standards related to V&V for solid mechanics, for fluid dynamics, for nuclear simulation. And a few years ago, they created a subcommittee called V&V 40, which is for computational modeling for medical devices. And after several years of work, they finally released a standard. So this is the first consensus standard that's ever been released regarding credibility of computational models for medical devices. Came out last year, and it provides a risk-informed framework for assessing computational models. It's not a quantitative framework, because it's much more generic than that. It just provides a kind of framework and a workflow for medical device industry to start thinking about how the different factors to consider when they evaluate medical computational modeling, and how to go about and it helps to kind of harmonize the approaches that's being done. So the main key features of the framework is this focus on the context of use. That's what COU stands for. So how you're actually going to use the model, and how specifically will the model be used to address a question of interest. And then the second part is model risk. So typically with computational models in healthcare, you will not only have different sources of you'll have different sources of information. So the model might be one source of information, but you might also be using animal experimental information, bench testing, clinical data. You might hedge against how your model is used a little bit by saying that the clinician should always use their experience. And then there's the consequence. Is it used for a very low consequence event, or will it, if you make a mistake, is the consequence something, some minor discomfort to a patient, or will the patient die? So we have these two axes, and the BNV40 framework basically asks users to kind of consider the influence and the consequence and evaluate the risk, and then basically use that risk to determine how rigorous they should be evaluating each of these other what are called credibility factors. So various factors related to what kind of verification is done, what kind of validation was done, including a focus on the comparator, and also something called applicability, which I'll talk about in the next slide. So the framework basically breaks down the credibility assessment into questions, important sub-questions, and it's a great start, but there's still a long way to go into making this really relevant to all the challenges that we face in medical devices and the kind of benefit-risk assessments that we have to make. One thing some work that I was doing a few years ago that is very related to the previous is on this concept of applicability analysis. So one of the main challenges with medical devices and using models in healthcare in general is you usually can't validate your model in the same way that you're going to use it. So contrast that to someone who develops a model of a crashing car, and they want to use it to maybe see how cars will crash as they change some design settings. They can do validation where they crash a car and really do very close validation, and then when they make changes and do the simulations for the prediction, it's not too different to the validation. Whereas you can't do that often for medical devices. So for example, this is a model of a stent, and and people have developed solid mechanics models of how this deforms, and how they validate it might be to put it into this machine, do radial loading, and look at the force displacement curve with hysteresis and see if the predictions from the model match. But then afterwards they'll simulate that in the patient with bending, with realistic loading conditions that the patient is undergoing, and across maybe a patient population, and that's very different to this. So how do we know that we can trust that kind of jump? So we have this issue: how relevant are the validation results to support the context of use? And very briefly, what we did in this paper was provide a step-by-step process to evaluate that question, and it kind of asked the user to think about 4 different settings: how the model, the validation experiments, the validation model, the context of use model, and the reality that we're trying to simulate. Think about what the differences are here, what the similarities are here, and it's kind of conceptually not that complicated, but then actually people find it difficult to apply. I don't really understand why, because it's not that difficult compared to the technical challenges. But we do find that when you go through this process, you end up identifying a lot of questions that wouldn't, oh, that's not good, that you wouldn't normally come across. So between those two, between the BNV40 framework and the applicability analysis, I'm not expecting you to read all these, but the idea is that we've started off with this very general question: how do we trust a computational model? And we can break that down into a set of questions. And one of those questions is, what is the uncertainty in the inputs, and how does this affect the uncertainty in the outputs? And that is something that's really being pushed at the moment, UQ, the BNV40 standard. And I want to emphasize that it's being pushed not in a nuanced way. So people appreciate that UQ is important. The BNV40 standard basically says that if the highest level rigor should correspond to comprehensive sensitivity analysis and uncertainty in all the inputs. There's a call for grants related to neurostimulation devices that also says if you're developing models, we want models of physiology that we can use to understand neurostimulation devices, and we would like uncertainty quantification in all component model parameters. Some of the work so the last few minutes I'm going to talk about modeling of some work related to cardiac models, where we're trying to get a little bit more information about how nuanced we should be when we make these kind of calls for UQ. So I'm coming at it from the idea that models can be used as clinical tools, and you will often have in those tools imaging data that will go into a model. There'll be some uncertainty associated with that. Steve did a great talk on uncertainty related to anatomy and data that you collect from the clinic. What often happens with these tools, though, is that you have a model in here, typically a cardiac cell model and things that are built using those, which have multiple internal parameters, and they're often highly uncertain. So ideally, we would like to have uncertainty in all these as well as uncertainty in these, and that will give you the uncertainty in the tool output. But how plausible is that? And so what I'm going to talk about in the next few slides is some work where we, to address that question, not with a commonly used cardiac cell model, which have 200 parameters, not individually named, not a clear link to physiological data. So there's no way that we could be characterizing the uncertainty in all those parameters. So what we did is developed a simplified model that has only it is a model of the canine action potential. It has it has representations of 6 important currents, but has certain judicious simplifications that allow us to only have 7 state variables and 36 parameters, which allowed us to, for the first time, kind of think about how this model would behave if we vary all those parameters simultaneously, because a lot of the work that's been done for cardiac UQ has focused only on, say, conductances, or and so initially I'm going to talk about some work, present some results related to arbitrary parameter variation, and then some empirically derived parameter uncertainty. So what we did initially was take all the parameters if they were physiologically constrained to be greater than 0, prescribe a log normal distribution. If they're otherwise, prescribe a normal distribution, and have this hyperparameter sigma, which is, you can consider like a knob that controls the total amount of uncertainty in all the parameters. So first of all, we apply a small amount of uncertainty to all the parameters and discover that the action potential is actually robust to a small amount of uncertainty, and that we didn't know if that was going to be the case initially, because cardiac models are kind of notoriously sensitive, and we see the same action potential shape. We were able to look at sensitivity indices and histograms of the outputs. But increasing the amount of uncertainty with this hyperparameter going from 1% to 3%, we start to get different behaviors, and as we go to 5%, we get lots of different behaviors. So then you have this problem that, and it's actually one of the reasons why UQ is not done comprehensively for cardiac models, is the models are going to fail, so what should you do afterwards? One approach is to kind of do a calibration where you say that all these parameters will keep only the ones that are good in some way, but there's the issue with that is that maybe some of these have failed because of some issue in the model form, the model structure of the equations, or maybe the parameter uncertainties that you prescribed are too wide, or maybe there's some correlation that we're not accounting for properly. So what we've been basically evaluating is a method called Monte Carlo filtering, which is a nice and very simple approach to identify which parameters are important regarding these different behaviors. So what we did was characterize the different behaviors into different types. So the green has no spike, the white has no, green has a spike but no dome, the yellow has no spike, the red action potentials are oscillatory. And by going through this process called Monte Carlo filtering, which don't have time to talk about, but it's basically a fairly straightforward process. We published this recently, so I'll have the citation at the end, which has more details. But it identifies 5 parameters that are highly influential in all of these behaviors. And to demonstrate that that was the case, if you fix those 5 parameters, and then vary all the others, you kind of remove almost all the other behaviors. And once you've identified that subspace in parameter space, then you can you can be more focused with your investigation and look for potential correlations. And I'll skip this slide, but it basically summarizes how by going through this process we can do more focused investigation and look at if these 4 parameters affect this behavior, do we expect that to be physiological reasonable, and if not, maybe we should be looking into the model form there and kind of iterating. And then that was all arbitrary parameter variation. So the next thing we did, because we have this 36 parameter model, and it's kind of feasible, it's hard work, but it's kind of feasible to come up with some kind of crude but empirically derived estimate of the real parameter variability in all of those. And it's going to be crude, and it's also going to neglect correlations, which is something that might be very important. But that's what we did. We've started to characterize, this is work in progress, but we started to try and characterize the uncertainty in all the parameters related to the IK1 current, do the same for the ITO current, and this is variability related to the population. So we have voltage climate experimental data where we do have data for each individual cell, so we can try and parameterize the model, parameterize the IK1 estimate the IK1 parameters for each individual cell, and then fit a distribution to that to estimate the variability, and then propagate that through the model. And this is just an illustration of the range of action potentials you get. We've been looking at histograms and other things, but it's easiest to just illustrate the range of action potentials for a presentation. And we've been doing that for these different currents, and you see that for the estimate that we have for ICAL, that's the one that gives us the greatest variability, and we'll try and do that for all the different currents, and we can go through the same process and identify which parameters are important, and this will allow us to maybe iterate on the model and eventually reduce the uncertainty. And the final thing we did, the final slide here, is we tried to look at a more useful output quantity of interest. So we kind of hypothesized that because we often don't care about the nominal behavior with cardiac models, we actually care about the relative change when you apply some perturbation, such as you apply drug block of the IKR current due to block based on a drug, that maybe that change in the action potential duration prolongation, this is the control experiment, or this is with the nominal parameter values, maybe this change, there wouldn't be much variability in that. Maybe all these action potentials would just shift across. That would be nice, but we didn't find that to be the case, unfortunately. We actually get quite a wide range in the distribution of the delta APD, even just for the variable, when you just look at the variability of one current. And obviously we're getting even more variability in the other currents. So to summarize, computational modeling is being used across a wide range of areas in healthcare now. Talked about 3 different application areas, and that leads to challenges in how we assess these models, and the community is starting to develop frameworks related to model evaluation. And one of the challenges is UQ, and we've started to investigate the feasibility of doing rigorous and fully comprehensive UQ. So the idea, maybe in 10 years down the line, we will have models that are like this, where we have inputs coming from the patient, and you characterize all the uncertainties in those, and you have a simplified model, and there's a uncertainty characterized in all those inputs due to the population, and then you get that uncertainty in the output. But at the moment, this work kind of elucidates some of those challenges. But the Monte Carlo filtering, for example, provides a path forward to improve new models. So that's the kind of information that we should be using in the development of new frameworks, and we should definitely be including that while there is uncertainty in the models, that should be included in our cost-benefit risk assessments, and at the same time, this kind of work will help create better models and modeling workflows. So thanks for your attention. I'd like to acknowledge some collaborators at FDA, and here's some references to some papers that the first one's going to come out in the next few weeks. So thank you, and I'm happy to take any questions.
Hi, thanks for the talk. That was really interesting. The thing that I was most interested in is the differences between the engineering approach and the medical approach to this kind of thing, because obviously you can't crash a person in the way you crash a car. But what I wanted to ask is it possible to use historical data from devices and things that have already been approved, and then go back in time and model them with our current modeling technology and say, well, yes, the technology is okay here, and we do have evidence from the medical side to show it?
So that's actually one thing that the...
Question, please.
Oh, okay. So it was a question about whether we can use, how we can use historical data in the evaluation of models and the development of models, or both?
To evaluate the models that we've got now and to get better ones.
Yeah, that's an area where there's another major difference between engineering and simulation, and healthcare. And not just historical data, but data that's being collected in the field, especially, I don't know what it's like in Europe, but in America there's registries and people are trying to collect what they call real-world data and real-world evidence that can be used to evaluate devices post-market. And so that's, we don't have a framework yet, but it's something that's being worked on to the extent that the BNV40 subcommittee, after developing the BNV40 standard that came out last year, has now formed 6 different working groups, and one is actually how do we use historical data as a comparator. So they're trying to think about what the challenges and the specific questions that should be asked about those, because we can use them, but there'll be differences to how you want to use the model. And yeah, they're working on it basically. David?
David Christini, Weill Cornell Medicine. Just sort of a broad FDA kind of question. In one of your very early slides, you had this chart of the various modeling modalities in the various modeling areas. And if I saw it correctly, I think I noticed that population modeling only had like 1 check compared, it seemed like it was not widely used.
Yeah.
That sort of surprises me.
Yeah,
It's of, like trying, because the work that you showed at the end is sort of related to the population of models in the cardiac cellular electrophysiology to try to see where a drug, or in this case a device, how to try to predict its efficacy over a broad type of range of people.
So I think one thing I should probably make clear with that is that a white square doesn't necessarily mean that there's no work being done. It's just no one's reported it yet, and we're going to continue collecting evidence. So I'm sure there's various places like that where we've collected that information. We're going to kind of propagate it, disseminate it to the FDA scientists, and I'm sure there'll be lots of places where people say, actually, no, we should have provided some feedback on how we use this in this center. So that's definitely one area. I think that there'll be a lot more work done. There is a lot more work done.
So the question you ask, when can we trust a computational model? I mean, and the way you described it, kind of that a model is produced and then you have a series of tools to help you understand whether you can trust it. But surely the fundamental question is, how do you build models that you can trust? And part of that trust, of course, is what is the claim for your model. Because if you claim that your model is right, well, it's not. It's simplified in all sorts of ways. So the modelers themselves need to be explicit about the claims that they're making, and the job of the people who are checking should check the rationale that they're using and the claims they're making. For example, you do an analysis based on varying a whole bunch of parameters, but these parameters are not real things. They are abstractions that turn up in a model. Yeah? So the notion of in what sense varying those parameters actually tells you something about a real-world system, that's a part of the job of the modelers. And it should be that the checker's job is to assess the credibility and scrutinise the arguments that are given. Of course, which partly means that all these modelling teams need more statisticians, which of course I would agree with.
Yeah.
But it does mean that the emphasis sort of happens in a different order. You know, the idea that the model is there and then they should pass it over to you, what do you think? It's just a crazy way of doing it, in my view.
Yeah, and I don't want to give the impression that, so the people at FDA scientists are developing models as well and working very closely in collaboration with academia. I mean, that's part of the reason why I'm here, right? So basically everyone has to work together as we develop the models and use the models, and for the modelers the focus will be on how do we develop models that we can trust, and for FDA it'll be how do we confirm that that's the case.
Let's thank Pras again, and then we'll move on next to Tiziana. Serena, if you want to start from